Hi, I'm Dr. Pierce, and welcome to this video on controlling the cell cycle. So one of the main tenets of cell theory states that all cells come from pre-existing cells. In other words, cells divide to form new cells. The next question that is inevitably arises, well, if that's true, where did the first cell come from? And that is a great question. That's one that's going to be answered a different day though, because in this video I want to explain what the cell cycle is, what happens at the various stages, and most importantly, how and why is it controlled. So let's first have a look at what the cell cycle actually is. When you follow cellular growth from a newly formed cell all the way to the point where it divides to form two genetically identical cells, you are looking at one complete cell cycle. That cell cycle can be divided, no pun intended there, into two sections called interphase and the mitotic phase. Interphase is further delineated into three phases called G1, S, and G2, while the mitotic phase is separated further into mitosis and cytokinesis. So let's have a quick boo at what occurs in these different chapters of the cell cycle. So looking first at the G1 phase, known as the first gap phase, this is a time of high metabolic activity. The cell is growing and therefore must be manufacturing all kinds of biological molecules. This results in high levels of transcription and translation in order to produce the enzymes necessary to get all this manufacturing done. Now, before we move on to the S phase, we must acknowledge the fact that cells are not constantly growing and dividing. Sometimes the cells are not ready or they're not needed to divide, so they enter what's called the G0 phase, which is a time of resting with respect to the cell cycle. They are still active and carrying out their functions. They're just not committed to division. Some cells enter into this phase permanently and become what is called terminally differentiated. They are what they are. They're not going to change or divide. However, other cells will temporarily enter the G0 phase and then return to the G1 phase when they receive signals that it is time to divide. Now, when the cells commit to divide, they must first enter the S phase or the synthesis phase, during which the DNA is replicated. This obviously needs to happen. If you're going to produce two cells from one, you're going to have to ensure that each gets a copy of the DNA. So that's what occurs here in the S phase. Then we enter the gap2 phase, where the cell continues to grow, but it also starts to manufacture new organelles and generally just prepares for the M phase, the mitotic phase that's coming up. So after all that effort, the cell is now ready to divide and enters the M phase, where the chromosomes will be divided equally, followed by the division of the cell cytoplasm and all of its contents. But before we get into the details of what happens in the M phase, it's probably prudent for us to take a quick trip down memory lane to remind ourselves the basic chromosome structure and some of the nomenclature that is associated with that. So here's your basic DNA double helix that makes up a chromosome. But when we are modeling mitosis and meiosis, for that matter, we tend to represent the chromosome like this. The green oval in the center there represents the centromere of the chromosome, which is a DNA sequence that will eventually facilitate the binding of the spindle fibers to the chromosome, and that's going to be necessary for chromosome division. Now, as mentioned earlier, during the S phase, the DNA is replicated, and when it is, the two DNA molecules are actually held together at the centromeres. At this point, despite the fact that it's still considered one chromosome, each of the two DNA strands are called sister chromatids. And it's important to show, as we zoom into a particular part of the chromatids, that they are indeed identical with respect to their DNA sequences. And that's good news, since each cell will eventually get one of those, and each of those cells needs to be the same. So let's take a look now at a nucleus that has two pairs of chromosomes, one long set and one short set. Note that there is one set that comes from the mother, the green ones, and one set that comes from the father, the red ones. And again, as mentioned during the S phase, the chromosomes are replicated, and they stay that way through G2 until the cell is ready to divide. So now it's time to move on to the M phase. It's worth noting here that the AP does not even require you to know the names of the individual phases of mitosis, but frankly, I don't think that's an onerous task, and it's a lot easier to grasp the important concepts of mitosis when you know the phases and you have a rudimentary understanding of what's going on in each phase. So we'll try and keep it simple. But let's pick up on the cell in late G2, and at this point you cannot see the chromosomes since they have not yet condensed into the tight structures. Prophase is initiated when the nuclear membrane disappears and the chromosomes begin to condense and become visible. Spindle fibers that grow from the two poles of the cell will now bind to the centromere of each chromosome. Unfortunately, I was not able to show that on this slide, but here you can see the process going on in a real cell. Now, once those spindle fibers bind to the centromeres, they help move the chromosomes to the center of the cell until they're aligned on what's called the metaphase plate. The spindle fibers hold the chromosomes there until they're ready to be divided. And you can see the chromosomes moving to the middle of the cell in this video. Now the spindle fibers are going to shorten, and the centromeres that were holding the two sister chromatids together are pulled apart. One of the two sister chromatids is pulled to each end of the cell, ensuring equal distribution of the genetic material. In telophase, the chromosomes actually reach the poles of the cell, and the nuclear membrane begins to reform. And at that point, the chromosomes begin to decondense once again and become invisible. Now, this video doesn't show all of that because sometimes the lines between the phases are not overly distinct and phases blend into one another. So for example, cytokinesis will often begin before telophase is complete. This is the division of the cytoplasm, and in animal cells it's initiated by the invagination of the cell membrane from both sides. This continues until the two ends meet and fuse, creating the two distinct cells. Now you can see in this video the cells have now separated and the nuclear membrane is rebuilt, and the chromosomes are now decondensing, all as part of late telophase. So now that the cell division is complete, we can zoom in on their nuclei to show that the two cells are indeed genetically identical to the original. And after looking at some of the details of the mitotic process, it would be wise to remind ourselves as to why this is actually important. The obvious answer out of the gate is that we need to grow. We're called multicellular organisms for a reason, and those multitudes of cells have to be generated, even if we're not all as cute as that baby. Another reason for mitosis is tissue repair. We're constantly replacing and fixing things, and for that we need new cells. And then there are the gazillions of bacteria that depend on mitosis, which is called binary fission in their case, to asexually reproduce. And this is all fine and dandy as long as the cells divide when they're supposed to and refrain from doing so when they're not. It should come as no surprise then that the cell cycle, like so many other biological processes, is tightly controlled. And if you need more convincing on the importance of that, consider what happens when the cell cycle control is lost. Cancer. Shown here is the result when mitosis and cell division goes unchecked and out of control. So let's talk a little bit about what that looks like, what that control looks like. Here is a cell cycle diagram again, but this time you can see that there are checkpoints at various places in the cell cycle. You have them at the end of G1, the end of G2, and somewhere in the middle of the M phase. So the cell is constantly having to make decisions as to whether or not to move on to the next phase. So why are these checkpoints at these positions in the cell cycle? To help us understand this a little bit, I would like to bring in an analogy of the SpaceX rocket mission. If you liken the G1 phase to the rocket being set up on the launch pad, it makes sense to have a major checkpoint here as decisions have to be made. If the systems are not in place or there are malfunctions that have to be fixed, the rocket needs to be taken off the launch pad and those issues need to be dealt with. And this would be the equivalent of G0. And the rocket could stay there for quite some time before the decision is made to launch, at which point it heads back to the launch pad. Now the reason why this is so important is because if you do decide to send the rocket up, you are committed to bringing it back down. So likewise, if a cell is going to enter the S phase and replicate its DNA, it is committing to cell division, and you simply cannot undo DNA replication. So you really need to be sure about it. Once the rocket is now in orbit, it's going to carry out its functions, but it will also be preparing for re-entry and landing. And again, before it does the latter, a systems check must be done to ensure that all mechanisms are working, just like the cell will use that time in G2 to ensure that the cell is ready for mitosis. Finally, the big moment arrives and the rocket makes a triumphant landing, hopefully, and heads back towards the launch pad, where it begins the process all over again. Of course, the only fault of this analogy is that you don't actually end up with 2 rockets out of this, and even Elon Musk can't solve that problem. I want to now move on to discuss briefly how the checkpoints work, and I will use the G2 checkpoint as an example. Cell cycle control uses 2 types of regulatory proteins. The first set is called CDKs, which stands for cyclin-dependent kinases. And since you know what kinases are, you can kind of guess what those proteins are going to do. The other set is simply called cyclins. It's important to realize that CDKs are produced and maintained at constant levels in the cell, much like the lac repressors, and they're also made in an inactive form, again, like the lac repressors. Since they are called cyclin-dependent kinases, you may have figured out already that they will be activated by cyclins. Cyclins, on the other hand, are named because of the fact that their levels in the cell rise and fall in coordination with the cell cycle. Let's pick this story up in G1. So the graph off to the left shows the relative levels of the proteins in the cell, and the model on the right shows the actual proteins inside the cell, where the orange squares represent the CDKs and the green triangles represent the cyclin proteins. Now remember that CDKs are maintained at a relatively constant level, while the cyclin concentrations will be quite low in G1. As the cell enters the S phase, the cyclin levels continue to rise while the CDK levels remain constant. This trend continues into G2, and you can see now that the cyclin concentrations are getting quite high in the cell. Eventually, a critical threshold of cyclin will be reached inside the cell. This will cause the cyclins to bind to CDK, and in doing so, they form a complex called the maturation promoting factor, or MPF. The rapid formation and resulting high concentration of MPF in the cell triggers the cell's passage past the G2 checkpoint and into the M phase by phosphorylating and activating proteins and enzymes that are necessary for carrying out mitosis and cell division. And as with all things controlled, what started must be stopped. So near the end of mitosis, the cyclin portion of MPF is destroyed, releasing the CDK portion and sending the concentrations of MPF and cyclin plummeting in the cell. This signals now the end of mitosis and the beginning of a new cell cycle. And the process now continues. Cyclin levels begin to rise again throughout the phases of G1, S, and G2 until MPF is formed, sending the cell into the M phase and cell division before returning once again to the G1 phase. So there you have it, the cell cycle, which can repeat over and over and over again. And as long as it stays under control, you really don't even know that it's going on at all. Thanks for watching.